Hello everyone, I'm ICSAE7 from 11 Maret University. Today I'd like to share my research in the topic in vitro fibroblast cell culture from Pelung chicken embryo and its potential application. Fibroblasts are the most common cells of connective tissue in animals. The cells are well known of its important role and capacity to regenerate skin structure. Nowadays, fibroblast cell culture technology has become a widely used tool for tackling various biological problems. Chicken fibroblast cell culture has demonstrated successful applications for cytotoxicity screenings of active compounds, for example, and then for wound healing assay or to elucidate pathogenesis in poultry as well as production of vaccines. Although some earlier studies have reported the establishment of cell culture derived from domestic animals in Indonesia, little was done to use local chicken as the source of primary cell culture. Furthermore, the availability of fibroblast cell culture from domestic chicken can serve as an alternative way for the preservation of the genetic resources in the long run. So in this study, we isolated fibroblast cells from Indonesian local chicken breed, that is Pelung chicken. This Pelung chicken was selected as a primary cell culture donor based on its inherent superior characteristics. The chicken is well known as a singing cockerel and resistant to highly pathogenic avian influenza virus due to the presence of antivirus genome expressed. And also, Pelung chicken was often used as superior breeding stock in poultry crossbreeding to increase productivity and improve the genetic quality of local chicken. First of all, I would like to explain briefly about wound healing assay, since we are going to mention about that later. Wound healing assay serves as a simple method for studying collective cell migration towards chemical stimuli released by the wounded area. And this method employed cell culture as an in vitro approach to study the wound healing process. This is also called a scratch assay, since it is done by making scratch on a cell monolayer and then capturing images at regular intervals by time-lapse microscope. Besides wound healing assay, the fibroblast cell culture is also versatile for conducting screenings regarding the safety of medicinal compounds such as Centella asiatica. As you might have known, Centella asiatica is a famous traditional medicinal herb with many health benefits. Several studies suggested acetic acid compound in Centella asiatica may help to accelerate the healing process of burn and scratch wounds. Another study also confirmed the influence of triterpenes of Centella asiatica on increasing the metabolism of lysine and proline. Those are amino acid building of the collagen molecule. So, while there is no doubt on its prominent beneficial effect, it is also important to examine the cytotoxicity of this herb in a normal cell. The objectives of this study were, first, to establish and characterize primary fibroblast cell culture from Pelung chicken, and second, to explore its application for downstream in vitro-based study. In this case, we would like to try it with wound healing assay and cytotoxicity tests against bioactive compounds of Centella asiatica leaf extracts. This is a summary of methods that we have used. We isolated the fibroblast cells using mechanical technique and then cultured them in a DMEM standard media supplemented with 10% of FBS, and all cells were then passaged routinely, and the cells used in the study were at least coming from passage 4 or so. And then the cell viability checking, we use trypan blue exclusion dye assay, and we observe the morphology of the cells in a native culture, but sometimes we also use markers in an immunostaining method. And for the wound healing assay, we have described in earlier slides, while for the treatment with extract of Centella asiatica, we will describe the following slides. From here, I would like to explain about the results. This figure shows the growth of Pelung fibroblast cells. And this is asterisk that marks the explant. Cells outgrowth from the skin explant adhere firmly to the dish surface by 24 hours after isolation, and the typical fibroblast-like cells exhibited a spindle-shaped structure that reached about 60 to 70% confluent at 72 hours and then covered all surface in 96 hours. As the cell density increased, you can see that the cells appears more aligned and parallel to each other. And what we obtained in this time range is normal if we compare to the regular technique of explant cell culture from skin. The cells appear as plump spindle-shaped or stellate-shaped cells with centrally placed oval or round nucleus. Occasionally, we found cells with two nucleus as shown in this image, and the stellate-shaped cells with multitude of cytoplasmic projections such as an active fibroblast. The cell growth was examined to check whether the culture conditions support healthy cell proliferation and development. The exponential phase lasts for 2 days with an average maximum cell number reach about 79,000 cells per centimeter square, and the number of fibroblast cells of Pelung chicken in the study is rather high as compared to the previously described in other chicken, but possibly because it's due to the difference in the species that we use and also the media used. And for the routine culture, we observe no contamination of mycoplasm. As you can see here, there is a very clear nucleus staining without cytoplasmic dots, as commonly seen when there is a mycoplasma contamination. And also the bacteria and fungi growth were also prevented by using antibiotic and also antifungal. As for the population doubling time, we see 184 hours, which is quite slow, but possibly we argue the cell population is not fully homogeneous yet in the first subculture, thus causing prolonged PDT. As we know that primary cell culture generally has lower growth characteristic and has a limited cell cycle compared to cell lines with a more homogeneous cell population, we also know from the previous report that which mentioned that after several times of passage, then they observe a shorter PDT when the cells becoming more homogeneous. So here we demonstrated a successful culture of Pelung fibroblast cells, and we can preserve it by using liquid nitrogen. So in the future, the cell stock can be used for other purpose of experiment. After we establish the fibroblast cell culture from Pelung chicken, we would like to check if it can be applied for wound healing assay. In wound healing process, fibroblast cells play a crucial role. The phase is divided into 3: inflammation, proliferation, and remodeling. During proliferation phase, fibroblast cells will migrate to the wound area. Cells will proliferate and then synthesize new extracellular matrix for culture of the wound area. The potential of Pelung chicken fibroblast cells to be used as a wound healing model in this study was based on the contractile and migratory nature of the cells. This figure shows the cells before scratching, and this is a panel of cells after scratch. The width of the wound area shows around 390 micrometer, which begins to narrow up to 245 micrometer after 4 hours, and then 4 hours later, or after 8 hours of the scratch, the width of the wound area was reduced by 68% to 122 micrometer. The process of closing of the wound area was almost complete at 24 hours, although some spaces between cells remained open. So this experiment proved that a Pelung embryo fibroblast cells culture has the migration ability to cover the wound or scratch area within 24 hours. Next, we would like to move to the application of the cell culture for cytotoxicity test. In vitro cell viability and cytotoxicity assay has been widely used for testing cytotoxicity of chemicals and screening of drugs. And Centella asiatica here was oven dried. We took the leaf parts and then oven dried, finally powdered, then macerated with 70% of ethanol for 24 hours with occasional stirring. And the mixture was then filtered using Waldman filter paper, and then the supernatant was evaporated using a rotary evaporator at 40 degrees Celsius to obtain extract or paste like this. And this extract was then diluted in an aqueous at twofold concentration to obtain the final concentration of 250, 500, 1000, 2000, and 4000 microgram per milliliter. In this study, the cytotoxicity of Centella asiatica extract was tested on Pelung chicken fibroblast cells. Fibroblast cells were treated with the Centella asiatica extract for 24 hours at various doses. The results show that the cell viability decreased in a dose-dependent manner, where the increase in the Centella asiatica dose resulted in lower cell viability. A dose of 250 microgram per milliliter of the extract caused 16% decrease in cell viability compared to the control, and cells treated with 1000 microgram per milliliter of the extract showed 51% cell viability decrease. The results obtained in this study were similar to the other Centella asiatica cytotoxicity test using MTT reduction assay on 3 kidney cell lines. Centella asiatica extract in the methanol solvent showed non-toxic concentration within the range of 100 to 250 microgram per milliliter, while the extract with distilled water showed LC50 above 200 microgram per milliliter. So this indicates that Pelung fibroblast cells as a primary cell culture showed similar sensitivity and response with cell lines, or at least if we compare to the 3 cell lines which I mentioned above. In conclusion, cells isolated from dermal skin of Pelung chicken embryo exhibited typical fibroblast cells, sigmoid growth curve, and survived long-term preservation. The fibroblast cell cultures from Pelung chicken responded well in the wound healing assay and showed cell viability in dose-dependent manner to the extract of Centella asiatica. Finally, I would like to thank Ms. Harlina Wulandari for data acquisition, Mrs. Nita Tikawati for the valuable discussion, Ms. Ika, Ms. Laila, Mrs. Santi for providing the extract. This work was supported by PNBB Research Grant of 11 Maret University. Thank you very much.